in the prealar vein. To do that, we have to go through the outer ear, the middle ear, the inner ear. When we're starting this process, the sound waves, pressure waves, are funneled into this external auditory means. And this auricle or pinna is our outer ear. That kind of acts like a funnel. Sometimes when we can't hear as well, we might cup our ear. If you've seen pictures of these old-timey ear trumpets, they'd have this like big funnel-like thing if you couldn't hear very well. That's kind of the idea. You need to gather the air pressure sound waves in and send it down the external auditory meatus. Now, we talked about yesterday the sound energy out here loses some energy due to resistance, impedance, by the time you get into the cochlea. But that we have three mechanisms to help ameliorate or decrease this energy loss. Without these mechanisms, less than 1% of the energy that would be out here would reach the cochlea. The three mechanisms again are, number one, this external auditory canal or meatus acts as a resonant column. So it amplifies, especially frequencies that we hear the best. Then, when the air pressure waves come in, they hit the tympanic membrane, the eardrum, and they cause it to vibrate. That vibration is determined by the frequency of the sound waves. Higher frequency are going to make it vibrate faster and lower frequency slower. The loudness or amplitude pressure, the higher the pressure, the more displacement of the tympanic membrane. That tympanic membrane is then attached to the three ossicles, the malleus, incus, and stapes. This is the second of our mechanical savings of energy, and that's the ossicles act as a lever, mechanical advantage. The stapes foot plate pistons into a little opening into the cochlea. That little opening is oval shaped, so it's a nice name called the oval window. So that pistoning of the stapes foot plate into the cochlea here sets up fluid waves in the cochlea. Because the tympanic membrane has a surface area which is effectively 17 times greater than the stapes foot plate, that's our third thing that causes us not to lose too much energy along the pathway. This shows you a different picture of it. Here's the tympanic membrane and the malleus, incus, and stapes. There's the stapes foot plate and the oval window there. So we're transducing this to get it to that cochlea. The middle ear, in addition to having these ossicles, has another opening called the auditory tube. This runs from our middle ear down to our pharynx, our throat. Old school, it was called the eustachian tube, but it's sort of trying to get away from proper names of people. So we generally call it the auditory tube now. The problem with calling it the auditory tube is people often mistake it for the external auditory canal or meatus, and it's different. This is a different tube and it has a completely different purpose. What is the purpose of the auditory tube? Well, we know that there's external pressure, atmospheric pressure, and we know that that pressure changes as we go up and down in altitude, for instance. Well, we need to equalize the pressure across the tympanic membrane. In other words, the pressure in the middle ear needs to equalize with the outer atmospheric pressure. The way that we do that is we can put air up and down through the auditory tube from the throat. And we can equalize the pressure. We can do this kind of manipulation with our jaw or moving this, and our ears will pop. Well, the ears popping is the equalization of pressure across our tympanic membrane. Sometimes our ears pop as we're going up and down Deer Park Road. As we're changing altitude and changing pressure, we have that mechanism. So the function of the auditory tube is to equalize the pressure across the tympanic membrane between the middle ear and the outer ear and external environment. One problem, oh yeah, Neil, go ahead.
I was just going to say that sometimes when I'm sick with like an upper respiratory tract infection, it's harder to get my ears to pop. Is that because of inflammation of the tube or congestion?
Exactly. That's really what the problem is with the auditory tube, is it provides a conduit for air to equalize the pressure, but it also provides a conduit for bacteria and viruses. So if you have a throat infection or something like that, it can sneak up the auditory tube to your middle ear. And it can cause a middle ear infection, which can cause an earache, but it can also cause more of a challenge to equalize the pressure, as you were mentioning. So that is sort of the downside of it. Some little kids, little children, get a lot of middle ear infections because this tube and everything hasn't fully developed. Sometimes they get so many middle ear infections that you take a tiny, very small tube and you actually punch it through the eardrum, the tympanic membrane, and you drain the excess fluid out down the ear. Now you don't want to do that too casually, but it's called tubing the ears. You can actually punch it through, and you'd say, well, doesn't that damage the tympanic membrane? Well, if it's small enough and then eventually you pull it out, it can heal up. You can rupture your entire eardrum. You can rip it and do things, but the tube is small enough that it doesn't do any permanent damage. So that's the function of the auditory tube. The ossicles are very small. These are the smallest bones of the body, as you would expect. And they ratchet back and forth, piston back and forth, and set up fluid waves in the cochlea. But because, of course, sound waves are pressure waves, they have energy. If the sound is too loud, they can potentially damage these ossicles. Too much energy. So we have some muscles. The pediis muscle is one of them, and the tensor tympani muscle is another. These muscles are reflexive. They're skeletal muscles, but they act reflexively, which is unusual for skeletal muscles. But what happens is, if you're aware of a loud sound, like somebody gonna shoot off a gun close by you, these muscles can reflexively contract and hold the ossicles to prevent them from moving too much and preventing their damage. Now, if somebody shoots the gun behind you and you're unaware, then there's no time for those muscles to react, and so you get the damage. If the sound is loud enough, it doesn't matter. If those muscles, you can overwhelm them and damage those ossicles and your conducting system in that way. Of course, as we age, these tympanic membrane and the ossicles and all of this can also get, through wear and tear, they don't work as well. And so that can lead to some loss of hearing problems. When we get to the cochlea, we have, this text, we'll look at it in a picture. The cochlea is a snail shape. It's got about 2.5 turns. In this diagram, it's been untwisted a bit just so we can see it a little bit easier. To get us oriented, here's our external auditory canal or meatus. Here's our eardrum or tympanic membrane. Here's our malleus, incus, and stapes, foot plate of the stapes. Here's our oval window. Here's a round window, which we'll also be talking about. And here's our cochlea. The cochlea, just like the vestibular apparatus, in the semicircular canals, it has an outer bony labyrinth, bony area with perilymph fluid in it, in this paler blue area, and it has an inner membranous labyrinth, and that has endolymph fluid in it. Each of these areas has a name. For instance, this area on this side, as you're going, is called the scala vestibuli. And on the far side, you also have perilymph, it's called the scala tympani. And in between, you have the cochlear duct or the scala media. The cochlear duct contains endolymph fluid, but it also contains the hearing apparatus. So that's right lined up along here. That hearing apparatus, by the way, is called the organ of Corti. The organ of Corti. And it's made up of hair cells and some membranes that we'll look at. Essentially, the hearing mechanism, as we have air pressure waves displacing the tympanic membrane and moving the three ossicles and pistoning that stapes foot plate into the oval window, you set up fluid waves in the scala vestibuli. That scala vestibuli fluid waves in the perilymph, if it's very low frequency that we can't hear, it will go all the way down and through an opening called the helicotrema and bypass the cochlear duct completely and go to the scala tympani. Fluid waves here are dampened by the fact that we have a round window that's flexible, so you don't get back waves, and you allow the fluid to move. Well, what about hearing? That's stuff we don't hear. In frequencies that we can hear, it displaces a membrane between the scala vestibuli and the cochlear duct. That's called the vestibular membrane, also called Reisner's membrane. But again, that's a proper name, so we don't use it too often. And if you displace it here, it will set up fluid waves in the endolymph, which will cause a displacement of hair cells in that area and can lead to more or fewer action potentials. This is a little animation of it, short one.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes the perilymph in the scala vestibuli to vibrate, which in turn causes displacement of the basilar membrane. Short wavelengths from high-pitched sounds cause displacement of the basilar membrane near the oval window. This movement is detected by hair cells of the spiral organ, which are not visible in the animation. Long wavelengths from low-pitched sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ, which are not visible in the animation. When the vibrations reach the perilymph in the scala tympani, they travel to the round window where they are dampened.
This shows it in a still form. The fluid waves are moving here, and they can move through the cochlear duct to the scala tympani and activate hair cells in that area. High-pitched frequency waves displace the membrane and move through closer to the oval window. closer to the base. Then intermediate and then low frequency are closer to the apex, to the helicotremon. Therefore, depending on the frequency of your waves, you're displacing and activating hair cells at different places along the length of the cochlea. So the brain will interpret where it's coming from in the cochlear duct and then encode for pitch in that way. Again, higher pitch closer to the oval window, intermediate pitch about midway, and lower frequency towards the apex or farther away. You can see that here. Here's high frequency vibrations, here's medium frequency, and here's low frequency. Now, of course, unless it's a totally pure note, you're going to get some displacement at multiple places simultaneously, and the brain will integrate that. So even if you play a chord of 3 notes, you're going to activate it in 3 different places at a minimum. And the brain will put that together. This is another simple diagram. Here's your external auditory canal or meatus. Here's the tympanic membrane, malleus symphysis stachys. Here's the auditory tube coming in. Here's the oval window and the round window, the perilymph, and it'll move through in this area. Here's our cranial nerve 8 taking the signals away. Now we're going to watch this animation in a minute, but let's finish this up and then we'll do a summary animation of it. Notice high frequency waves displace closer to the base, and then here's medium frequency, and then there's low frequency. Our best, where we hear, the frequency that we hear the best is about 2 to 4,000 hertz cycles per second, by the way. This is our most, we hear that frequency the best. 20,000 is our upper range, really, of hearing. Now, inside of this scala medium, the cochlear duct, we have the organ of Corti. Just to get us oriented, this is our snail-like structure, and we've done a cross-section through it. We've cut right through the snail. Going back, we've cut right through. We've taken a cut right through there. Now, when we're looking at that, here we see the scala vestibuli. That's where the fluid waves originate from the oval window pistoning into it. This is the perilymph fluid in here. Here's our vestibular membrane, or Reisner's membrane. And here's our cochlear duct with endolymph fluid in here. When fluid waves get generated in the endolymph of the scala vestibuli, they displace this vestibular membrane, push it into this cochlear duct, and that causes fluid waves to happen in the endolymph of the cochlear duct, and then it passes on through to the scala tympani, the perilymph fluid, and goes to the round window. But in the meantime, you have this apparatus, the organ of Corti here, that has some hair cells, and sticking out from the hair cells, you have stereocilia and kinocilia. They stick out here, and they attach to a membrane above the hair cells called the tectorial membrane. This is the membrane above. Then the hair cells are also attached to a membrane below the hair cells, and that's called the basilar membrane at the base. When you have fluid movement, it causes a shearing action as the basilar movement, as the basilar membrane shears back and forth in one direction or another. Now, if it turns so that the stereocilia are bent towards the kinocilium, you open more gates, you get a more depolarization, and you get more action potentials. If it moves it so it's going the opposite way, the stereocilia are moving away from the kinocilium, then you close gates, you get a relative hyperpolarization, and you get fewer action potentials. We want to remember, here's the stereocilia and the kinocilia. We want to remember that in this microenvironment, potassium ions are high outside of the cells. So for instance, outside of the hair cells here. And so when we open the gates, they're potassium gates. They're mechanically controlled by that shearing motion. And if you go towards the kinocilium, you're going to open more potassium gates. Potassium will come in to the hair cell. That depolarization will open more voltage-regulated calcium gates at the base of the cell, and that will cause the release of more neurotransmitter. Glutamate is the neurotransmitter, and that will bind to cranial nerve 8 and result in a higher frequency of action potentials. If the movement caused the stereocilia to move in the opposite direction of the kinocilium, you would close more of these potassium gates, you would get a relative hyperpolarization, you would have less calcium coming in, and you would release less glutamate, and you'd have fewer action potentials in cranial nerve 8. At rest, when they're all sticking straight up, the stereocilia and kinocilium, you would have a few gates open, potassium, and a few action potentials. So we can move it one way or another, and that's how we send signals. Do you feel okay about that, or do we want to chat about it more? Sneak it past you here. Well, this is a nice animation, and it sort of gives the whole package of it. It's somewhat lengthy, but I think it's worth seeing. It's not that long.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
So hopefully that helps you visualize what's going on in there and seeing all that mechanism. This is not to memorize again, this is just to give you a kind of a reference to see about decibel levels of common things. Notice above 85 decibels, 8 hours exposure causes hearing damage. So you have to be careful that if you're working around any kind of machinery that is greater than 85 for 8 hours, then you're going to have hearing damage unless you wear hearing protection. Here, if you're above 100 decibels, you certainly want to do it less than 15 minutes if it's unprotected. Chainsaw, more than a minute can result in permanent hearing loss. How many people go out and use a chainsaw and don't use any hearing protection? Should always use some hearing protection. You have these over-the-ear ones and the others. So that's important. Obviously, loud music. Shotgun blast, you're not where it's coming, especially. So you can see that jet takeoff within 75 feet, you're going to rupture your tympanic membrane. That's not funny. That's a lot of energy. But what about hearing impairments? We can categorize hearing impairments in two main subcategories. One is conduction deafness. This is the most common form of deafness of aging. As we get older, we lose our hearing over time. We become hearing impaired at least. And this is a wearing out of the transmission mechanism, often to the oval window, the tympanic membrane, the ossicles, that kind of thing. It impacts all frequencies, but it is helped by hearing aids. If you increase the ability to the amplification, hearing aids can be helpful with this. You've probably heard that Apple is coming out with it. They're going to use their device for hearing and hook it up to an iPhone or what have you. That'll be interesting to see how well that works, but theoretically that could be a good addition to the ones that are already available. The next type is sensorineural, or just simply perception deafness. This impacts the hair cells and the nerve, the cranial nerve 8, if these get damaged. Well, what could damage these? Well, this perceptive deafness, it's the most common form of congenital deafness. You're born with some hearing apparatus that's not working correctly. The hair cells or the nerve is not done well. But there are certain drugs that can affect and cause perceptive deafness. Certain very strong antibiotics, very necessary, but there's, if you get a bacterial resistant disease and you have to prescribe one of these strong antibiotics to knock it out, to save the patient, a side effect could be it could damage their hair cells and their nerve and cause perceptive deafness. And very strong painkillers, if you use them too long, can cause this. So a lot of people that become addicted to painkillers or something, they can experience perceptive deafness over time. How do we fix this? Hearing aids won't work for perceptive deafness, but you can do cochlear implants. And you can actually put a mechanical device in there, an electronic device, and that will stimulate cranial nerve 8, replacing the hair cells essentially. And so that's how you treat that. Interesting. Anything we want to chat about hearing before we move to vision? Feel okay about that? Okay, well let's move to vision. This will be the last of our sensory, and of course it's the most complicated. Vision is, the energy form is a type of electromagnetic radiation that is approximately between 400 and 700 nanometers. visible light. The reason we call it visible light is that our photoreceptors, our rods and our cones, are able to respond to wavelengths within this range. So we see that. Within this visible light range, we actually have the rainbow spectrum, all the colors, don't we? We see it as white light, but if we use a prism or whatever, we can divide it out. And so that the lowest, the shortest wavelength and the highest energy are the purple-blue ray wavelengths. And then we go to the green and the yellow and the oranges and the red. Past that, going shorter wavelengths, we have ultraviolet light. We know that that can give us a sunburn. It has energy, doesn't it? From the sun, X-rays, gamma rays. Going the other way, we have infrared, microwaves, radio waves. Longer and longer wavelengths and lower energy. They're inversely related. So we have these. One of the things we're concerned about is how much energy blue light has. I think we talked about that earlier this quarter for when we're talking about sleep. When we're looking at devices that emit a lot of blue wavelength, it might be somewhat more damaging to our eyes. We might be more susceptible to cataracts at an earlier age. And we certainly might have a harder time sleeping if we're doing that too late at night. So that shows us that electromagnetic spectrum and that portion of it. Well, to understand vision transduction, we have to look a little bit at the anatomy of the eye. Here's our eye with our eyelids and eyelashes and all that. Here we see the front of the eye. The white part of the eye is called the sclera, and that comes all the way around the eyeball. But at the part right from here to here, the sclera becomes the transparent cornea. Transparent because, of course, light has to pass through that. The hole in the middle of the eye, the opening, is called the pupil, and that can change its size, the aperture. The aperture of the pupil, to allow more light or less light in, is controlled by some smooth muscles, and this makes up the iris, the colored part of the eye. So we can close the pupil aperture or open it up more. As light comes in through the cornea, it comes into this anterior chamber region, and there's some aqueous humor, some fluid here, and then it comes and it strikes the lens. The lens is connected to some suspensory ligaments which connected to a muscle called the ciliary muscle. This allows the ciliary muscle, allows the lens to change its shape. It can become more convex or more flat. The lens, by being able to change its shape, can change the refraction of the light, the bending of the light, so that you can look at faraway objects or near objects in a process called accommodation. What I mean by that is if you're looking far away, the lens stays fairly flat and it doesn't refract the light very much. If you're looking at a close-up object, the lens will become more convex, change its shape, refract the light more, and therefore you can focus on this closer up object. After moving through the lens, you go through the vitreous humor in the posterior chamber, and you focus the light right here on the retina at the back of the eye. We really have three main layers. The outer fibrous layer, often called the corneal scleral layer, is this cornea and continues on as the white part of the eye as the sclera. Then we have the choroid layer, and that is a very vascular layer and has some pigment behind it. And then we finally have the innermost layer, which is the neural layer, commonly called the retina. And that's where our photoreceptors and other neuronal cells are found. If we look at the back of the eye, we see the macula lutea. That's called the yellow spot. That's what that means in Latin, I believe. The macula lutea. And in the middle of that macula lutea, that yellow spot, we have the foveus centralis, which is a little depression. That is the central axis of our eye, and we have only cones in the foveus centralis. That's the place of greatest visual acuity. Where the optic nerve exits the back of the eyeball, we call that the optic disc or the blind spot. There's no photoreceptors where the optic nerve is exiting, and that's why it's called our blind spot. If we want to detect our blind spot, we can look ahead and put a finger like this, and as we move, somewhere out here we should lose our finger. We don't see anymore. That's our blind spot. That's all. There's no photoreceptors there, so we can't see objects in our blind spot. This shows you an actual eyeball that's been cut very thin and then put on a slide. We're looking at the opposite orientation. This is our cornea, and the sclera comes around, and here's the lens, but it's kind of broken up and all that. Here's our anterior chamber, our posterior chamber. Here it is with some labeling. Anterior chamber with aqueous humor. Anterior and posterior chamber are here, and together they can be called the anterior cavity. The posterior chamber filled with vitreous humor or vitreous body. We secrete a fluid into the aqueous humor, into the anterior posterior chamber, and it circulates around. It drains out of an area called the channel of Schlemm. This is our lens. It's attached by zonula fibers to the ciliary body, the ciliary muscle, so it can change shape. And around the pupil, right here's the pupil, the opening, we have the iris, which is the colored part of the eye, and can change the opening aperture of the pupil. There's our optic nerve. We were talking about refraction briefly. Refraction is when light passes from one medium to another, if those two media have a different index of refraction, then you will bend the light. We're quite familiar of this because when we look into a pond with fish, we know by experience pretty quickly that the fish isn't actually where he appears to be, correct? If you try to stab him and you don't account for that refraction, you're going to miss and you won't get your dinner. The fish is not quite where he looks like because of the bending of the light, the refraction. Well, when we go from the air, which has a refractive index that we've arbitrarily set to 1, the refractive index of the cornea is 1.38. So that change in the index of refraction causes the light waves to bend. Now there are really 4 parts of the eye that refract the light. The cornea refracts it the most, but it's not adjustable unless you do a surgery. So the cornea bends it the most. Then the aqueous humor bends it just a little bit, the fluid that it passes through next. Then the lens bends it, but the lens can change its refractive ability because it can change its shape. And finally, the vitreous body or vitreous humor refracts it just slightly. Ideally, we want to focus the light right on the retina, right on our photoreceptors in the retina. Anything that interferes with that causes a blurred image and causes problems. How do we change the lens shape and therefore its refractive ability? Well, the ciliary muscle is a round muscle, like is shown in this diagram, around the lens, and it's connected to the lens by these suspensory ligaments. Here they are. If the ciliary muscle is not contracted, it will be holding these suspensory ligaments tight, and the lens will be flat. If you want to change the shape of the lens, the ciliary muscle can contract inward towards the center of the lens, loosening the suspensory ligaments so they're not as tight, allowing the lens to become more convex and changing its ability to refract. Here it is with the ciliary muscle relaxed. The lens is in its flattened orientation. We would want this orientation when we're looking at a distant object, because we don't want to bend the light very much if we're looking at something far away. However, when we want to look at a nearer object, the ciliary muscle can contract inward towards the lens, loosening the suspensory ligaments, allowing the lens to become fatter and more convex, and bending or refracting the light to a greater degree so we can focus on a near object. This change is called accommodation, and you can see the word here. It allows it to happen. Now, I'll mention this again on Friday, but I just want to say very quickly while we're talking about the lens, and we'll remind you about it. The lens becomes less flexible as we get older, and we're not able to change its shape as easily and accommodate, which is why you need reading glasses or whatever. This loss of flexibility of the lens, where you could no longer focus as well on near objects, you have to pull them out further, what have you, or wear glasses, is called presbyopia. So that's a loss of flexibility of the lens. On Friday, we'll pick up a number of the disorders of the eye, and we'll look at several of them to see what's causing, and then we'll keep going. And of course, we want to get to how we're eventually going to transduce the light images into receptor potentials and then eventually action potential patterns. And that gets a little bit complicated in vision. So that's what we'll do on Friday. Remember, we don't have any lab tomorrow. There's no lab tomorrow. Turn in your lab report.